So, ladies and gentlemen, I'm very much pleasure to introduce my research work. The title is Reconstruction of a transmembrane protein, tetraspanin, CD9, into lipid bilayer by introduction of ganglioside GM3 and tetraspanin. So this is a slideshow. The left first report is agonine, as indicates the slide. The left is composed of lipid and carbohydrate and some proteins. And that function is the platform of some signal transduction. For example, stress, antibodies, adhesion, bacteria, and toxin. But one question is still remaining. How is the enzyme recruited? Yeah, because the outer membrane of left, it is very clear that many ligands link to the corresponding receptors. How is the signal transduction on inner membrane of left? For example, autophosphorylation enzyme or cutting enzyme of signal peptide. But I said already, how is the enzyme recruited in the inner membrane? So, for my research work, there is a very important technique. Two techniques is important. First one is this one, Langmuir-Burgett technique. So, lipids solution was spirited on the subphase using micro syringe. After evaporation of the solvent, the monolayer was compressed at 50 centimeters square per minute, very slowly. The half side of cell membrane can be obtained. And this slide shows us by using lipids. First one is a dipalmitoyl phosphatidylcholine. Second one is diureoyl phosphatidylcholine. The third one is dipalmitoyl phosphatidylethanolamine. And second technique is atomic force microscopy. Atomic force between probe and sample is able to measure. Some value of atomic force means same distance. Same value of atomic force means same distance. So we can get the map, height map of the construct. So this slide shows the GM1, ganglioside GM1, the structure of GM1. It's a characteristic ganglioside GM1 has a thiaryl alpha 2,3 galactose residue. And this is the other group's research. So GM1 composed effect of surface pressure on the AFM image for GM1, DBPC, DOPC, 22929 monolayer. Surface pressure is so. A conclusion, the percolation pattern in the GM1, DPPC, DOPC monolayer changed as the surface pressure was varied. And then, this is my work. about the ganglioside GM1 alpha left. But GM1 alpha is not commercially available. We must synthesize it. GM1 alpha in DOPC, DPPC monolayers and hybrid bilayers are prepared. Distribution of GM1 alpha is investigated by AFM. So this is the structure of GM1 alpha. Alpha series ganglioside containing a sialyl alpha 2,6-N-acyl galactosamine residue is characteristic and localized on cholinergic neurons exclusively. So this is a chemical synthesis of the GM1 alpha. Compound 1, very shortly I'll introduce. Compound 1 was synthesized through 6 steps from galactosamine. And compound 2 was synthesized through 7 steps from lactose. And compound 7 synthesized through 4 steps from galactose. And such component was linked so to get the tetrasaccharide. And then compound 10, this is sialyl derivatives, was synthesized through 9 steps from galactosamine. And ceramide part 14, compound 14, was synthesized through 7 steps from galactose. So total steps as 45. Maybe it takes 2 years. But we got the GM1 alpha. So first we can make a monolayer. Surface pressure versus area per molecule isotherms is shown here. So it makes a complete monolayer. Then AFM image for GM1 alpha, DOPC, DPPC 12929 monolayer. A is 15 millinewton per meter. It's a surface pressure. It means a low surface pressure and more higher. Pressure is increased. So first image indicate a small domain is associating the more bigger domains. And then big domain, in big domain, GM1 is located around of the domain. And then GM alpha left is depart from the domain. We can see the white part is the ganglioside left. And then last one indicates the ganglioside left is again associated. So such change was very clear. And this slide shows subphase is changed. Subphase changed into 157 millimolar sodium chloride. So the conclusion is almost same, but it looks like round. So, yeah, but for monolayer experiment, sugar chain was vented on the mica plate. So difference of the height is smaller. It is not clear. So we change the bilayer, indicates the right side. First layer is a DPPE, ethanolamine, and second layer is a sample. Sample means that GM1 alpha, DOPC, DPPC, 12929. So in this case, sugar chain is real size, real height we can detect. So it's very clear, and the contrast is very clear. So its conclusion, self-assembly by surface pressure. GM1 alpha, DOPC, DPPC, ternary monolayer, and hybrid bilayer obviously shows GM1 alpha raft. The raft was associated, separated, and associated again according to increase of surface pressure. The association of raft causes enzyme localization on signal transduction. This is my former part. And second, reconstruction of transmembrane protein. So first reported, one group reported, but their artificial interaction added. So it means histidine tag aquaporin Z and nickel chelate lipids. It's an artificial compound. But they can deconstruct the aquaporin Z. This is our interest. GM3, CD9, CD81 complex. This complex regulated cell growth and proliferation. The complex regulates also cancer cell proliferation. So without artificial interaction, we measure. So first, we have to make a monolayer. So this is a pi A, surface pressure versus area per molecule isotherms. So it indicates the complex monolayer. And then FM image. We can get also, first one is 10 nanomillimeter millinewton per meter, and second one is 20 millinewton per meter. It increases the surface pressure, compaction. So first one, 9 images we can detect in the 9 observed point. The second one, we can get 9 images from the observed 9 point. Also, third one, 8 images from 9 points. Last one, 9 images from 9 points. It means a common phenomenon. And then cross-sectional study of raft. The height, we can easily understand the height. So, about 1 nanometer height was observed. All case, all surface pressure, we can detect the 1 nanometer height. So this means a monolayer. So also carbohydrate part was vented. So height is very small, only 1 nanometer. then a hybrid bilayer, 10 nanometer, 10 millinewton per meter, and 20 millinewton per meter. 6 images we can get from 9 observed points. Second one, 7 images we can get from 9 observed points. 30 millinewton per meter, 6 images we can get from 9 observed points. The last one, 5 images from 9 points. And then cross-sectional study of raft. So it indicated more higher results. One part is 6.3 nanometer, and ganglioside raft indicated 11.9 nanometer. So about 7 nanometer and 12 nanometer. Okay. So it means the sugar part is remaining. So we can observe 12 nanometer. And the 7 nanometer means the DPPC rich domain. It's higher than DOPC because DOPC's legs is shorter than DPPC. And then reconstruction of tetraspanin. The experiment is the hybrid bilayer was kept in CD9 solution 2 microgram per milliliter for 5 minutes. So 5 images we can get from 9 observed points. And that height was changed. And second part is different surface pressure, 40 millinewton per meter. The 2 images from 9 observed points were no change, but 7 images among 9 observed points were changed. And cross-sectional study of raft reconstructed by tetraspanin. So difference is the underlined number. 16 heights. 16 heights indicates the reconstruction of the tetraspanin. So it means 3 nanometer increased. And second part, 40 pressure, higher pressure, also 16 nanometer was observed. So it means the tetraspanin was reconstructed. So that's all my data. Thank you very much for kind attention. What?
The total yield.
Yield.
Yield. How much that you get out if you start with 100 grams or whatever? What's the yield?
Yield.
After so many steps of chemical synthesis, very complicated.
I got only 3 milligrams last step.
And you started with how much material?
Maybe 10 kilograms.
Okay.
I want to thank you for this talk, because for many years I tried to understand how lipid rafts were activated. And I'm thinking here in terms of the T cell receptor interacting with the MHC class 1 or class 2 in terms of activating. There is an immunologic synapse involving adhesion molecules. So it acts as a suction cup, bringing the two cells together, the macrophage and the T cell together for signaling. It goes through the lipid raft. So that gives me an indication, maybe because of that suction, cells coming together gives you that surface pressure to activate the lipid rafts. And here you've done the study on this aspect that could give an answer to why these lipid rafts are working, which is really, really interesting. I thank you very much for that.
Thank you.
I still like to comment on this. I mean, it's very important what you did here, and these numbers are important, but this is still an artificial system. So in the living cell, you have lateral pressure. I don't know how big it is, but it's not varying this much. Yeah? And therefore, you have a steady state situation. And there has been work with fluorescent labeled proteins, membrane proteins, and fluorescent labeled lipids, using this STAT method by Hell in Göttingen, who got the Nobel Prize for this 2 years ago or something like this. And I know, since we participated in the study, that in the rafts in living cells, in the plasma membrane of a living cell under the microscope, in average has a lifetime of 10 milliseconds and a size of 10 nanometers. So that was between cholesterol and the sphingolipid, sphingomyelin or GM1, and it was unclear whether a protein takes part in this or not. So just the lipids. But they are very short-lived in the living cell. And the size in this study was very small too. Diameter of 10 nanometers allows only 2, 3, or 4 molecules, not more.
But if you have the cells coming together,
then this is a different situation. Then you have a protein-made microdomain. So these are membrane contact sites we have within the cell, for example, between the ER and the plasma membrane, ER Golgi, ER trans-Golgi network, ER lysosome, endosomes, ER peroxisomes, and so on. And they have a constant different distance, but they are mainly constructed by proteins which bind to both sides. These are not rafts as such, these are microdomains, yeah? Or nerve endings, postsynaptic membrane, presynaptic membrane. This is not raft, it's not just lipid-lipid. Raft is only lipid-lipid interaction. That's the difference. Microdomains are known for a long time. The synaptic domain. Yeah, but these are the definitions. Yeah. And there are annular lipids, and so on. Yeah. So I have one question. You synthesized this GM1 alpha, yeah, and showed the properties. This has the sialic acid alpha 2,6. Yeah. Now, if you compare this with the normal GM1, which has alpha 2,3, what would be the difference?
What?
In the behavior?
I don't know.
You don't know. But this would be important in
order
to know why what's the reason to have, in addition to alpha 2,3, the alpha 2,6 linkage.
Yeah, but biologically it's very important.
Yeah.
It only exists in the cholinergic neurons.
Okay.
Yeah.